Hello everyone. Our lesson for today is about microbial photosynthesis. So in this lesson, we will discuss more on how energy is being produced from utilizing light energy. You'll also learn the different factors and molecules that are involved in each pathway, how these processes are initiated, and which microorganisms undergo these pathways. At the end of this lesson, you will be able to identify microbial photosynthetic apparatus and differentiate the different types of microbial photosynthetic processes. So the motivation question for this lesson is this: how independent are we on light energy? So I hope you will be able to answer that one as we finish this lesson. So in photosynthetic microorganisms, there are three types of light harvesting systems. So basic types of photosynthesis include oxygenic photosynthesis, anoxygenic photosynthesis, and carotenoid-based system that is using bacterial rhodopsin. So anoxygenic photosynthetic organisms, they are believed to have evolved first. And chlorophyll-based photosynthesis is just an early event. Chlorophylls are tetrapyrroles, not like a hemi group. Magnesium replaces iron in the tetrapyrrole, and bacteriodopsins are carotenoids in halophilic archaea. So the energy for photosynthesis derives from the photoexcitation of a light-absorbing pigment. Okay, so photoexcitation leads to photolysis, or the light-driven separation of an electron donor molecule, such as water or hydrogen sulfide. Each electron is then transferred to an electron transport system. So you're maybe familiar with the electron transport system because we have already discussed this in a previous session on microbial respiration. We've said that there are three ways on how to generate ATP in a microbial cell. So it could be through substrate level phosphorylation, respiration linked phosphorylation, and photophosphorylation. So photosynthesis is actually a way for some of the microorganisms not to generate energy, to generate ATP, because they also have their own electron transport system. So the ETS generates a proton potential and the reduced coenzyme NADPH instead of NADH, as we've seen in microbial respiration. So instead of NADH, they produce NADPH for photosynthesis. The proton potential drives the synthesis of ATP through the F1FO ATP synthase. So what are the features of photosynthetic microorganisms? So they have light gathering systems, electron transport chains, ATP generating system, and an electron donating system to generate NADPH or NADPH. Okay, so the light gathering system example are the chlorophyll. So each type of chlorophyll contains a characteristic chromophore, light absorbing electron carrier. I know you're already familiar with photosynthesis because it has already been discussed during your botany subject, but we will be talking more of a microbial way of photosynthesis. Okay, so I hope you will be able to compare this one to that of the plant base, because as we know, the chloroplast in the plant cells are even evolved from cyanobacteria. Okay, so we will be able to compare that one later. So each type of chlorophyll contains a characteristic chromophore, which is the light-absorbing electron carrier. So chlorophyll molecules differ slightly in their substituent groups around the ring. Okay, these slight differences alter the absorption spectra of a particular type of chlorophyll. So chlorophylls have tetrapyrroles. So the phytyl side chain provides Now, a hydrophobic tail that anchors the chlorophyll to membrane protein complexes is just the site of the light reactions. Okay. So antenna complex and reaction center are all parts of the photosynthetic apparatus. To maximize light absorption, the molecules of chlorophyll are grouped together in an antenna complex. So these antenna complexes are arranged like a satellite dish within the plane of the membrane in an elaborate cluster around accessory protein. So the clusters then associate in a ring around the reaction center. So the protein complex in which chlorophyll photoexcitation connects to the ATS. So you have here different chlorophylls, carotenoids, phycocyanin, and phycoylated that are found in the antenna complex and the reaction center. So if this is the photosynthetic reaction center, these are some of the pigments that harvest light energy and directs the light energy into the reaction center. Now, different groups of microorganisms, photosynthetic microorganisms, have different types of electron donors and electron acceptors in the ATS. So the groups of photosynthetic microorganisms are the purple non-sulfur bacteria, green sulfur bacteria, cyanobacteria 1, cyanobacteria 2, and heliobacteria. No, at least for the type of electron donors and electron acceptors that they have. So for purple non-sulfur bacteria, they basically have bacterial chlorophyll A and B. Well, for green sulfur bacteria, they have bacterial chlorophyll C, D, and A. Chlorophyll A and both chlorophyll A for photosystem 1 and 2, and bacterial chlorophyll G for heliobacteria. And these are the different types of electron acceptor that are present in these different groups of photosynthetic organisms. Carotenoids are an important part of the photosynthetic apparatus. They are long-chain hydrocarbons with many double bonds. They provide light harvesting capacity in the 400 to 550 nanometer range of the visible light and also accept highly toxic singlet-state oxygen, which is generated during photolysis of water. Okay. So how does photosynthesis occur? So I know you already know this, but it should be better if you're going to review this. The primary photochemical site is the photosynthetic reaction center, the PRC. This is a protein chlorophyll complex within the cell membrane of microbial cells. Okay, absorption of a photon by a chlorophyll molecule results in a distribution of electrons in the tetrapyrrole. So that it becomes because of the photons, the once stable state of the electrons becomes excited. Okay, so the excited state has more energy than the initial, okay, or the ground state. So excitation energy is transferred to neighboring molecules by resonance transfer. So as the electrons are transported from one electron acceptor to another, the energy is also transmitted. Okay. So bacterial chlorophyll absorbs in the range of 700 nanometers, okay, of the visible light. This permits growth at lower depths in water, but doesn't have enough potential to extract electrons from water. So in early evolution, it was believed that sources of electrons were limited to H2 and H2S only. So they undergo actually anoxic photosynthesis because at the present times, especially for terrestrial organisms or terrestrial plants, the main source of electron is the water. So they photolyze the water to yield the electrons, protons, and oxygen as a byproduct. Okay. So, oxygenic photosynthesis is actually one of the earliest form of photosynthesis that occur on Earth. So the light gathering, electron transport, and ATP generating systems are all positioned together on the cell membrane. So remember, this is a bacterial or microbial cell. So many species have morphological adaptation that enlarge the membrane surface, to allow for more reaction centers. Okay, just like this, stacks or the lamellae. So different, no, types of organisms have different arrangement, of their chlorophyll or the photosynthetic apparatus depending on their need actually. So there are 5 groups of photosynthetic bacteria that are recognized. The first group is called the purple sulfur non-sulfur bacteria. The second is the purple sulfur bacteria. The third, the green sulfur bacteria. Fourth, the heliobacteria. The fifth are the cyanobacteria. How do they differ? Okay, so we could differentiate them based on the source of reducing power or the source of the electrons, the oxidized product, the source of carbon, and the heterotrophic growth, if they are capable of heterotrophy. So they're photosynthetic, but are they capable of heterotrophy? Okay. So purple non-sulfur bacteria uses H2 and reduced organic compound source of reducing power. They use oxidized organic as oxidized product, carbon dioxide or organic as source of carbon, and common for purple non-sulfur bacteria to be or to have heterotrophic growth as well. Purple sulfur bacteria and green sulfur bacteria have used both H2S as source of electrons. The oxidized product is a SO42 minus the source of carbon is carbon dioxide, and they can do limited heterotrophy. For cyanobacteria, the source of electron is water, the oxidized product is oxygen, and the source of carbon is carbon dioxide, and they also have limited heterotrophic growth. Heliobacteria, on the other hand, is very different because they use lactate and organic compounds as a source of their electrons. The oxidized product is an oxidized organic compounds. The source of carbon is lactate and pyruvate, and they are required, it's required for them to have heterotrophic growth. So first, let's focus first on the purple non-sulfur bacteria. So they are found on the surface of water that are rich in organic matter with low sulfide, because they are non-sulfur bacteria. Example are the Rhodobacter and the Rhodopsin-based. So for example, the reaction center in Rhodopsin-based viridis is consists of 4 molecules of bacterial chlorophyll, 2 molecules of bacterial phenopatin, 1 molecule of menaquinone, 1 molecule of ubiquinone, and 1 non-heme ion. So these are found at the reaction center. Cyclic photophosphorylation is the one that is done by non-sulfur bacteria. It will generate ATP, but no reducing power that is but photoreduction of NAD occurs through reversal of ATP synthase. So this is the photosynthesis in purple non-sulfur bacteria. The reaction center has bacteriochlorophyll 870, meaning the maximum absorption is at 870 nanometers. This is a series of electron acceptors. And in the process, ATP is produced. So during cyclic photosynthesis, meaning the electron is just recycled. The source of electron, the bacteriochlorophyll A, as it is being excited, the electrons comes out and it loses the energy, it will just go back to the bacteriochlorophyll A, the process. So during cyclic photosynthesis, this is what is happening. Light induces transfer of an electron from A bacteriochlorophyll 870 to bacteriophytopatin. Then the electron goes to ubiquinones, this one, the one that we are showing here. The ubiquinons QA and QB then to the ubiquinon pole in the membrane to form QH2. The flow to the cytochrome BC one complex results in the movement of protons across the membrane. An electron then flows to cytochrome C2 in the periplasm. From there, it is donated back to bacterial chlorophyll 870 to complete the cycle and regenerate the initial state. So it is called cyclic phosphorylation or photosynthesis because the electron is recycled back. There is also photosystem number 2 in purple non-sulfur bacteria. These are especially found in alpha proteobacteria. Purple non-sulfur bacteria. They separate an electron from bacterial chlorophyll itself. Electrons are then transferred to an ETS in the process, and ultimately an electron is turned to bacterial chlorophyll. So this process, which generates the ATP, is called cyclic photophosphorylation. PS2, unlike photosystem 1, provides no direct way to make NADH or NADPH for reductive photosynthesis. So if you notice here, this is the cyclic electron flow. Okay, so the electron donated by the bacterial chlorophyll, okay, at this photosystem is just recycled, but it is capable of heterotrophic growth. So lactate and pyruvate could be a source now of electron for the photosynthesis to occur, or the reducing compounds or the energized electron carrier molecules could be those that are produced from the TCA cycle. Okay. So this is another representation of the photosynthesis in purple non-sulfur bacteria. How the bacterial chlorophyll here is providing the electron, and these are accepted by these different ubiquinone, and passed on until it will lose its energy and will go back to that reaction center. So H2 can be used, or ethanol, isobutanol, succinate, and malate as a substrate. Growth in anaerobiosis in the presence of light causes photophosphorylation. Growth in aerobic conditions will yield heterotrophy and oxygen respiration. The presence of oxygen shifts to heterotrophic growth for purple non-sulfur bacteria. Photoreduction of NOD through reversal of ADP synthase and generation of PMF is possible. So the purple sulfur bacteria, the one that we discussed, are the purple non-sulfur bacteria. Purple sulfur bacteria is almost the same. Instead that they have PRC, or the photosynthetic reaction centers, that is distinct from other photosynthetic bacteria. So they can use H2S as an electron donor for an oxygenic photosynthesis and can accumulate sulfur granules in the cell. The third type is the green sulfur bacteria, which is otherwise called photosystem 1. So they utilize reduced sulfur compounds as electron donor in photosynthesis. Light reactions result in the formation of a strong reductant. The reductant can directly reduce NADP. Antenna pigments, bacterial chlorophyll A and C, and bacterial pheopitin A, high density of pigments in the reaction centers. They live in lower density aquatic environments. When reduced sulfur is present at those depths, there is low light intensity. For photosynthesis in green sulfur bacteria, they have a photosynthetic reaction center that is distinct from other photosynthetic bacteria. They can use H2S as an electron donor for an oxygenic photosynthesis and accumulate elemental sulfur granules in the cell. So green sulfur bacteria appear green and utilize reduced sulfur compounds as electron donors. So light reaction at the reaction center, which is bacterial chlorophyll 840, different from purple non-sulfur because it is bacterial chlorophyll 870. So the results in a strong stable reductant. They permit non-cyclic light-driven reduction of NADP without reverse electron flow. So this is the cyclic electron flow in green sulfur bacteria. Yeah, the reaction center is bacteria chlorophyll 840 that would uh, release become a source of electron. If you're a series of electron acceptors, okay, and when it loses its energy, it will go back to bacterial chloride 884. But for non-cyclic electron flow, the final electron acceptor is NADP plus that becomes reduced into NADPH. But the to replenish the lost electron, S2O3 and S2 could be a source of electron. Okay, this is another representation of the photosystem 1 with bacterial chlorophyll 814 green sulfur bacteria and how electrons are uh, flown from the reaction center to the different electron acceptor. So in green sulfur bacteria, the antenna pigments are packed into vesicles called chlorosomes on the inside of the cytoplasmic membrane. The chlorosomes are packed with bacterial chlorophyll C and are attached to the cell membrane by a sub-antenna of bacterial chlorophyll A in a structure called the base plate. Excitation energy passes through the sub-antenna to the reaction center. So this is the structure of the bacterial chlorophyll in green sulfur bacteria. So they appear as rods, 10 nanometer in diameter, lipid-rich structures containing the antenna, pigments, carotenoids, and bacterial chlorophyll C. You have the reaction center that is embedded in the membrane. The photochemistry of green sulfur bacteria is similar to photosystem 1 in cyanobacteria and plants. ATP is generated through a cyclic photophosphorylation and reduced pyridine nucleotides are produced by the non-cyclic FT reductase FMR system. So in the case of the heliobacteria, heliobacteria are gram-positive microbes that are commonly found in soil. So they have a unique bacterial chlorophyll, G, which contains a vinyl group, as is present in plant chlorophyll A. Bacterial chlorophyll G absorbs maximally at 788 nanometers, a wavelength that is not absorbed by other photosynthetic bacteria. Their carbon sources are simple monomers. While in the last group, the cyanobacteria are formerly called the blue-green algae. So they undergo oxygenic photosynthesis. That means they actually produce oxygen in the process as a byproduct. They appeared on Earth around 2.5 billion years ago, and there are two independent light reactions, the photosystem 1 and the photosystem 2, which are believed to be the ancestors of plant photosynthesis. The photosystem 1 contains the reaction center P700 that can reduce NADP. Photosystem 2 contains the reaction center P680, which splits the water via the process called photolysis. So they undergo oxygenic Z-pathway of photosynthesis, just like in plant cells. I know you are already familiar with the Z-pathway. So these are found in cyanobacteria and in chloroplast of plant cells. These include homologs of PS1 and PS2. 8 photons are absorbed and 2 electron pairs are removed from 2 molecules of water, ultimately producing oxygen. So oxygenic photosynthesis forms 3 ATP, 2 pH per water photolyzed and oxygen that is produced. So the ATP and pH are used to fix carbon dioxide into biomass in the dark reactions of photosynthesis. So photosynthesis in cyanobacteria follows this Z scheme. So I know you already know this. So as photons are absorbed by the chlorophyll at P160 680 photosystem 2. Now the electron is donated, it becomes excited and donated through the ATS, okay, which produces ATP in the process. As it loses its energy of the electron, it will be re-excited at P700 photosystem 1. It becomes excited again and it will lose the energy as it is passed on to via the different electron acceptors, and finally it will be taken up by NAD P plus and it becomes NAD PH together with protons. So in photolysis, 4 photons are required to split into 2 waters of oxygen. So I mean 2 molecules of water. Water is bound to a light-sensitive enzyme in the S system. 4 electrons are released, and the protons are released into the medium, and oxygen is generated. So in cyanobacteria, this is what occurs. This is another representation of the Z pathway of the photosynthesis, not just like in plant cells, and another representation, the Z pathway. Okay. This is how ATP is produced during the light reactions, because of the as the electron is donated through the electron transport system. So cyanobacteria antenna pigments are arranged in multi-protein complexes called the phycobilisms. Light energy is absorbed by the phycobilism na is passed to photosystem 2. So this is a photosystem 1 and 2. We have here the allophycocyanin and some accessory pigments. Photosystem 2 is a site of photolysis, the P680. So photolysis is the cleavage of water and the production of oxygen. So a photon excites chlorophyll P680 to 680, that meaning it has already released its electron. So the electron is passed to plastokinones and other electron carriers, and the last electron carrier is plastocyanin, which is a copper-containing protein. Finally, it arrives at chlorophyll P780, driving ATP formation, or the one we call the photophosphorylation. Photosystem 1, the P700 absorbs a photon and gets a higher energy. So it gives its electron to Fs centers. So these are a series of electron acceptors. And the last electron acceptor is the NAD+. So the antenna pigments are phycocyanin and phycoerythrin. Okay, so phycocyanin absorbs at 630 nanometers, or phycoerythrin at 550 nanometers. Some cyanobacteria can respond to ultralight conditions by synthesizing different pigments, which is what we call a process called chromatic adaptations. In chromatic adaptations, when cells are grown in green light, the rods are predominantly composed of the red pigment phycoerythrin. But when they are grown in red light, the rods are predominantly made up of the blue pigment phycocyanin. Okay? So both phycoerythrin and phycocyanin are open chain tetrapyrroles. Okay, so phototrophy is the process of harnessing of photoexcited electrons to power cell growth. Okay, so in most ecosystems, photosynthesis is based on chlorophyll. However, many prokaryotes use a simpler, more ancient form of phototrophy. So bacteriorhodopsin is a single protein light-driven proton pump that is found in halophilic archaea. The homologue is the proteorhodopsin that is found in marine proteobacteria. Okay, so in halophilic archaea, okay, so they live in high salt concentrations. So halobacterium halobium is the one that is best investigated, halophilic archaea. They are obligate aerobe. When oxygen concentration decrease, they will synthesize purple patches. So these provide a light-driven proton ejection system. So the patches contain the protein bacteriodopsin. The protein is attached to a molecule of retinal or vitamin A aldehyde. Bacteriodopsin is similar to the visual pigments of animals. Bacteriodopsin absorbs light at 570 nanometers and becomes bleached. It then rejects the proton to the exterior of the membrane. The deprotonated bacteria rhodopsin then takes a proton from the cytoplasm. So to maximize light absorption, Halobacterium salinarium and Archaea pack their entire cell membrane with bacteriodopsin. So the protein forms trimers that pack in hexagonal arrays forming the purple patches, not the purple membrane. Okay, so this is another representation of how phototrophy occurs in halophilic archaea. Okay. So as we end our lesson for today, let me give you these summary questions. What is photosynthesis and what are the different types of photosynthetic process in microorganisms? So I hope you will be able to really differentiate these different types of photosynthesis in microorganisms. Okay, and the different apparatus that they use, the different electron donors and the electron acceptors. Okay. So let me thank again Miss Maria Shalita Esrasal for the help in making the slide that I used in my presentation. If you have questions about this lesson, you can contact me through my email or your lecturer through the email provided, their email provided to you. Or you can key in your questions at our official Facebook Messenger group chat. So thank you very much and see you again in our next lesson.